this is Eric from Stanford University, and this is the first video in a three-part series on fungal disease, and the topic is the classification of pathologic fungi. The second video in this series will discuss the specific clinical manifestations and diseases of fungal infections, and the last will cover antifungal medications. The specific learning objectives of this video are to describe the general characteristics of fungi, including cellular structure and microscopic appearance, and to list the 5 primary categories of pathologic fungi, including representative species within each. Let me first ask, what exactly is a fungus? In common usage, most laypeople would suggest these organisms, mushrooms. Perhaps some would point out also that mold on an old piece of fruit is also a form of fungus. But the fungal kingdom also includes things that we don't necessarily think about initially as being fungi. For example, you know all that blue stuff in blue cheese? Guess what it is? That's right, fungus, specifically a genus of mold called Penicillium, which fascinatingly is the same mold genus that's responsible for penicillin. In addition, you know that yeast that we use to make bread rise and to brew beer? Also fungus. And even miso paste is produced in part by a species of Aspergillus. So there's a few examples of fungi, but how do we define it more precisely? In the most general sense, a fungus is any organism that is eukaryotic, has a rigid cell wall containing molecules called glucans and chitin, and are incapable of producing food. That is, they, along with animals, are heterotrophs, as opposed to organisms such as plants that can produce their own food from an external, non-organic energy source, and which are called autotrophs. Their cellular structure is actually very important in both defining what a fungus is, as well as understanding the mechanisms used by antifungal medications. First, like all cells, they have a cell membrane composed of a phospholipid bilayer. However, instead of that bilayer incorporating cholesterol, like animals do, it incorporates a different sterol called ergosterol. Just outside of the bilayer exists a thin layer of chitin. If chitin sounds familiar to you, that's because you learned about it in middle school biology as the polymer of N-acetylglucosamine that provides the strength and rigidity to the exoskeleton of insects and crustaceans. Next is a crisscrossing network of glucans, which are polysaccharides composed of glucose monomers. Glucans include glycogen, which animals use to store energy, as well as cellulose, which is found in plant cell walls. In fungal cell walls, the predominant glucan is beta-1,3-glucan, represented here in red. And this is interwoven with other glucans, which vary between fungal species. Finally, anchored within this polysaccharide matrix are various glycoproteins. When we discuss the cell membrane of a fungus, we're referring only to the phospholipid bilayer. The cell wall refers to everything external to the membrane. A question both interesting and instructive comes up once in a while regarding whether fungi are more closely related to plants or animals. The obvious answer to most people is, of course, plants. But let's take a closer look at that comparison. For plants, animal, and fungi, are they unicellular or multicellular? Both plants and animals are always multicellular, but fungi can actually be either. Which of these have a cell wall? Plants and fungi. Does the cell membrane contain sterols? No for plants, but yes to animals and fungi. Do organisms in the kingdoms employ chitin in their biology? No for plants. As mentioned a moment ago, some animals do, and all fungi do. Which of these display locomotion? Neither plants nor fungi do, while almost all animals do. And I already mentioned their respective nutritional models. Plants are autotrophs, producing their own food, while animals and fungi are heterotrophs, which require autotrophs for the production of food. Finally, one more line. Can they infect humans and cause pathologic disease? No for plants, but yes to both animals and fungi. So looking at this chart, it might seem like more of an even split as to whether fungi are more like plants or animals. However, recent genetic analysis has determined that despite the conventional wisdom to the contrary, fungi are more closely related to animals than to plants. I'm going to now start discussing the specific biological categories of pathologic fungi. The most important distinction to make is that between yeast and molds. Yeast are typically unicellular, though some species can occasionally organize into filamentous structures called hyphae and pseudohyphae, of which we'll see a picture in a minute. Molds are multicellular and are typically organized into hyphae. Yeasts are usually white in color. Molds, as you may have personally observed in either your refrigerator or bathroom, can exist in a wide variety of colors. Yeast reproduction is typically by an asexual process called budding. Mold reproduction is typically through spore formation, which can be either sexual or asexual. And finally, yeast forms smooth, round colonies when cultured in the lab, while mold colonies appear fuzzy. The single most clinically relevant fungal group is a yeast genus called Candida. Candida contains multiple species which cause disease in humans. By far the most common is called C. albicans. This is a commensal organism normally present in the oropharynx, GI tract, and vagina. It causes opportunistic infections in immunocompromised states, or when the normal bacterial flora is disrupted by, for example, antibiotic use. Candidal infections can show up almost anywhere in the body, as we'll see in the next video. There are 4 other important, though much less common, candidal species. They are glabrata, Krusei, Parapsilosis, and Tropicalis. In some ways, glabrata is the odd one out, as it tends to be physically smaller than the others, and phylogenetic studies indicate that it is more closely related to other non-candidal yeast species. It's very possible that the next decade will see the organisms currently known as Candida glabrata reassigned to a different genus and possibly given a new name. The other medically important yeast genus is Cryptococcus, which also causes opportunistic infections, particularly meningitis and pneumonia. It has 2 species which cause disease in humans. First, the more common of the 2 is Cryptococcus neoformans. This is found in soil samples throughout the world and is most commonly associated with birds, particularly pigeons and chickens, as well as rotting vegetation. The less common species is Cryptococcus gattii, which I believe is the correct pronunciation. This is found predominantly in tropical and subtropical regions, though has just recently emerged in the Pacific Northwest, particularly Vancouver Island. It's believed to be associated with exposure not to animals, but rather to specific tree species. Let's move on to molds. Although they all seem to look more or less about the same macroscopically to the eye, they are actually an extremely diverse group of organisms. The vast majority of them are actually non-pathologic. Most pathologic species fall into 2 specific groups, the genus Aspergillus and the order Mucorales, which includes genera Mucor, Rhizopus, and Rhizomucor. Both mold groups and their spores are found in soil and decaying vegetation throughout the world, and both can cause a particularly aggressive and devastating rhinosinusitis, as well as various forms of pulmonary infection in immunocompromised patients. An important distinguishing characteristic of molds, which deserves a little bit more attention, is their microscopic structure. As mentioned earlier, molds are predominantly composed of hyphae, which are long, filamentous branching structures. Hyphae in some species contain septa, which are observable internal cross walls, while others do not. Another microscopic feature of molds is conidia, which are the clustered spores responsible for most asexual reproduction and are the primary vector for humans to become infected. A cluster of conidia is attached to the hyphae via a stalk called a conidiophore. Different species of mold can be largely identified by their specific morphology of these microscopic structures. For example, compare the upper mold to this one. Notice how the conidiophore-conidia complex looks completely different. That variation between species can be helpful in identifying these in the microlab. So we've talked about yeast, and we've talked about molds. What's left? The next major category are the dimorphic fungi, sometimes referred to as endemic fungi. And what makes them distinct is that they can act as either yeast or molds. They typically exist in the environment as a mold, where they are found in the soil. Then if that soil gets disturbed and the spores are sent up into the air, they can be inhaled into a person's lungs, where they grow as encapsulated yeast. As you might imagine, since the means of human infection by most dimorphic fungi requires inhalation, the most common manifestation are pulmonary infections, particularly subacute ones, which can develop over days to weeks rather than over hours to days. Something else about the dimorphic fungi, which is why they're sometimes referred to as endemic fungi, is that they're very region-specific. So while Candida and Aspergillus are really found throughout the world, examples of dimorphic fungi are really found concentrated in very specific geographic locations. Some examples include histoplasma, located in the American Midwest and South. This organism is particularly fond of areas frequented by birds and bats. Birds and bats aren't necessarily reservoirs themselves, but their droppings can apparently alter the soil chemistry to make it favorable for histoplasma growth. Other examples include blastomyces, also found in the American Midwest and South, as well as a bit in Canada. Coccidioides, which is a little bit of a tongue twister, and most people refer to as just cocci, of which there are actually two main pathologic species, is found in the American Southwest. Paracoccidioides, located in Central and South America, which is more likely to lead to a much more chronic infection than the other fungi on this list. And Sporothrix schenkii, which is the one exception to the geographic specificity of these organisms because this is found throughout the world. And this is the one dimorphic fungus which is also more commonly acquired via the skin rather than in inhalation through the lungs. The next major category of fungi are the dermatophytes. These are a collection of fungi which all share the ability to metabolize and subsist on keratin, which is the key structural protein in human skin. Dermatophytes cause superficial infections only, and these are typically named after the part of the body involved. For example, a dermatophyte infection of the torso is called tinea corporis, corporis for body, and I'll be discussing all of the various dermatophyte infections in the next video. Identification of the exact species is usually unnecessary for diagnosis and treatment, and in fact, I would guess that excluding dermatologists, not one doctor in 100 could even name a single specific dermatophyte species off the top of his or her head. Dermatophyte infections are typically only a cosmetic problem, but they can be surprisingly difficult to eradicate. The last of the pathologic fungi to discuss is Pneumocystis. The taxonomy of Pneumocystis is a source of continued confusion among doctors. It was originally believed to be a protozoan parasite in humans and rats, at which time it was named Pneumocystis carinii. It was determined to actually be a fungus in the 1980s, and the human pathogen was later renamed Pneumocystis urobetsii once it was evident that it was a separate species from the rat pathogen. For historical reasons related to nomenclature battles, urobetsii is frequently yet probably technically incorrectly spelled with just one I at the very end. However, people still refer to this as Pneumocystis carinii not infrequently. Partly this has been perpetuated by the insistence of holding on to the acronym PCP. PCP is the name given to a clinically distinct form of pneumonia caused by Pneumocystis. It originally stood for Pneumocystis carinii pneumonia, but even after the species was renamed, clinicians did not want to change the name of the disease, so now they claim it stands for just Pneumocystis pneumonia, which in my opinion is just a bit silly. Another relevant characteristic of Pneumocystis is that because of differences in biochemistry and cellular structure compared to other fungi, Pneumocystis infections are treated with antibacterial antibiotics and not antifungals, which further contributes to the confusion regarding taxonomy. In addition to the major types of pathologic fungi, there are some bacteria which masquerade as fungi, specifically Nocardia and Actinomyces. Both of these develop filamentous branching structures visible with microscopy, and they cause subacute and chronic infections of the lungs, central nervous system, and soft tissue, most commonly seen in immunocompromised patients. Infections by nocardia and actinomyces can be clinically indistinguishable from fungal disease, except for the fact that, as with Pneumocystis, they are only susceptible to antibacterial antibiotics and not to antifungals. So that's it. To quickly summarize, there are 5 major clinical categories of pathologic fungi. The yeast, which include Candida, of which C. albicans is the most common species, and Cryptococcus. The molds, which include Aspergillus and Mucorales. The dimorphic fungi, including Histoplasma, Blastomyces, Coccidioides, Paracoccidioides, and Sporothrix. The dermatophytes, the individual species of which do not matter. And finally, Pneumocystis, a single species which gets its own category because it's quite clinically distinct from the other fungi listed here. And please note that this classification scheme is more of a practical scheme based on their clinical manifestations and is not meant to be an accurate representation of their formal taxonomy. I hope you enjoyed the video. The next in this short series will cover the various clinical manifestations of fungal infections.